Hello, everyone. My name is Pam Soltis from the University of Florida, and it's my pleasure to welcome you to the Plant and Algal Genomics session. Plant genomes are amazing in their diversity, from the small streamlined genomes, as in the bladderworts or lentibulariaceae, to huge complex genomes that may have undergone multiple rounds of whole genome duplication that may be packed with repetitive sequences or both. And this diversity provides the basis for addressing fascinating questions, but also presents numerous challenges for assembly and annotation. Our presentations today will address both some of these questions and also the challenges. We'll have 5 speakers in this session, 3 10-minute talks and 2 5-minute talks. Each talk will be followed by a short Q&A. When we get to the point for the Q&A, please type your questions into the spot provided on the portal. And also, if you like a question that's been asked by someone else, please so indicate by clicking on the thumbs up. This will help me prioritize the questions for discussion. At this point, I'd like to introduce our first talk, which is by Jim Liebensmack from the University of Georgia in the US. And the title is The Open Green Genomes Initiative, Comparative Investigation of Plant Genome Content and Structure Across the Land-Plant Tree of Life.
Hello, everyone, and thank you for inviting me to speak in this conference. It's wonderful. I'm going to be talking with you today about this Open Green Genomes Initiative, which is a work that's been funded through JGI's Community Science Program and highly supported by the team at Hudson Alpha and JGI, and I will acknowledge some of them on our way through the talk. I also need to acknowledge the collaborators on this project. We have nearly 100 folks that have agreed to contribute either material or knowledge throughout this project. So this was the proposal that went in had 99 PIs on it, and quite a few are participating. Now, one thing I need to tell you about plant genomes is that they're rather complex. I mean, this crowd should know this, but just for others at the conference, plant genomes are very complex. The genome sizes can vary quite a bit, and the number of genes in the genome can be quite large relative to human or other vertebrate genomes. And what we are really, this is quite a challenge as we try to understand the way in which these genes are interacting with each other and their evolution following the genome duplications and gene duplications that have given rise to this larger number of genes. Now, how do we go about this? Well, we take an approach that our old friend Charles Darwin first started thinking about as he was cruising around the world on Beagle and jotting trees in his notebook. Jump ahead now to today, and we're at a point where we're in the phylogenomics age. We're using genome scale data to resolve phylogenetic relationships, and then using those phylogenies to understand gene family and genome evolution. So last year, about this time, a little bit later, the 1000 transcriptomes initiative paper came out, where we were using transcriptomes from over 1,000 species arrayed across the Viridiplantae, the green plant phylogeny, to understand their relationships and aspects of genome and gene family evolution. That was all done with transcriptomes. Now the 10KP project, which you may have been hearing about from Pam today, and the Open Green Genome Initiative are attempting to generate whole genome sequences for species across the green plant phylogeny. In the case of the Open Green Genomes, we're really focused on land plants, and we're really focused on making high quality reference grade chromosomal genome assemblies. What I'm showing here in blue are some of the texts that were originally proposed for sequencing in the Green Genomes project. In gold are some that other projects have been generating, and in black and red are other genomes that the Joint Genome Institute have been been generating, analyzing, putting into Phytosome, a database which many of you may be aware of. So the primary objectives of the Open Green Genomes are to improve our understanding of genome structure and gene family evolution, and particularly with regard to the Department of Energy's mission. So looking at genes and gene families involved in that are relevant for bioenergy production or remediate carbon acquisition and remediation of habitats. And how we do this, we really want to fill in the gaps that in the phylogeny of available genome sequences. So some of our sampling rationale includes phylogenetic position. I just mentioned this, we're going to fill in the gaps. Genome size and complexity. We're going to have to sequence some very large genomes, and we in fact are working on, and I'll talk a little bit about some very large genomes, the complex genomes that we are sequencing. But if we have a choice between choosing a highly heterozygous octoploid or a diploid, inbred diploid, we're going to choose the inbred diploid for a particular lineage. And then experimental tractability is very important because we want people to be able to use, experimentalists to be able to use these genome resources to drive their own research. And one case where we're doing that right now is with sea-fern. Sea-fern is a model for homosporous ferns. Some of you may have worked with sea-fern gametophytes, one shown here in an intro biology or intro plant biology course. And so we knew ahead of time, based on genome size estimates based on flow cytometry, that the genome size is rather large, over 10 gigabases per gamete genome. Well, what we started with is just a shotgun sequences, short read sequences, and did K-mer analyses on this. This is work done by folks at Hudson Alpha, associated with JGI, Jeremy Schmutz, Jane Grimwood, and their teams. And we see here is that we've got, again, a genome size that we're estimating a little bit under what the reported genome size, largely made up of repeats and low heterozygosity. In fact, this we believe is a fully homozygous reference organism that we're looking at. And we're taking advantage of the biology of homosporous ferns. So homosporous, what does that mean? That means that one spore makes a gametophyte. That gametophyte can make both male and female gametes that can join and form a sporophyte. If you use male and female gametes to derive from the same gametophyte, then that's going to give us a fully homozygous sporophyte. And so we've used that data, taken almost 96 of these fully homozygous gametophytes, or I should say double haploid sporophytes, to make a genetic map for the genome. And one thing that's very interesting about this is that we really don't see across this genetic map much evidence for a segregation distortion. There's pretty uniform, it seems that there's pretty uniform recombination throughout this nearly 10 gigabase genome. Now, this is work that, again, a lot of the sequencing and the assembly analysis is being done by the JGI team led by Jeremy Schmutz, but the material and the support in terms of understanding the biology of sea-fern is coming from Blaine Merchant, Zhonghua Chen, Doug and Pam Soltis are involved, and others. So this genome is about to be put into Phytosome, and we're working on a genome assembly for, or a paper to describe the assembly and the biology of the genome. Another place that we've been putting from our early effort is to better understand monocot genome evolution. And so we've added, we have in the OGG quite a few monocots that, portions of the monocot that are representing lineage in the monocot phylogeny, for which there is currently no genome available. And so this is really important, particularly since when you think about how our cereal grains are all in the grass family: rice, wheat, maize. Our major cereal grains are all in the grass family, Poaceae within the monocots. And something that's been interesting as people have been sequencing and analyze some of these genomes is the importance of whole genome duplication event throughout monocot history and the footprints of the genome duplication events in some of the model grass genomes that have been sequenced. So this is work that came out a couple of years ago in genome biology from Patterson Lab, and they're showing that quite a few of the genes that they're looking at in these genomes are derived from duplicates of those genes are derived from whole genome duplication and tandem duplications as well. And so to better understand, where these genes are coming from and what's happening following with evolution of function, for instance, following genome and gene duplications, we need to fill in the holes, better understand, get some more representatives of lineages within the monocots that have not been sampled. So one of the first genomes that we've assembled and analyzed is a chorus, which is the sister lineage to all of the other monocots. And so we now again have a chromosomal assembly for a chorus americanus. This is also being placed in phytosome. And David Sankoff and his team, collaborators, have been using that genome together with other available monocot genomes to make ancestral reconstructions of these genome sequences. And something very interesting to see is we can take that and those ancestral reconstructions for the genome, paint them onto the extant genomes, and see how blocks are duplicated and have been rearranged and so on. Focusing more within the grasses, we've added ferrous, which is, if we go down and look here, we see ferrous is a grass that is outside of the core Poaceae, including all of the grain genomes. And so if we compare ferrous to rice, they're fairly collinear, as I'll show in a second. And in order to better understand what's been going on within the grasses in terms of genome evolution, up to now we've been using pineapple, ananas, as a outgroup for analyses of our grass genomes. And you can see that ananas has got quite a few rearrangements relative to these grass genomes. And some of them were following this whole genome duplication, so-called row genome duplication, the common ancestor of all grasses are represented here. So we now generated a Joinvillea genome and started analyzing it relative to these grass genomes. And you can see that there are far fewer rearrangements, but we still see footprints of these genome duplications. So for instance, here we can see that there's a segment of this Joinvillea genome that is lining up with two separate regions on chromosome 1 and 4 of the ferrous genome, and similarly the rice genome. So these genomes together, that is Joinvillea and ferrous, are really improving our understanding of the ancestral genome, both before and after this whole genome duplication event that occurred in common ancestor of rice, rice, wheat, and maize. So I'll leave it there and be happy to answer any questions you may have about the project. Thanks so much.
Thanks very much, Jim. We'll take time for just one question, I think, after your presentation. And at this point, I guess I'd like to ask you, what's on tap for next? Do you have a large number of genomes that you'll be sequencing? And you've already accomplished some of these. What are some of the things we can be looking for in the near future?
Well, you can look for these genomes that I talked about in my presentation to be coming out in publications in the coming year. Look for the data to be coming out in phytosome and before the end of the year. But some other things that other genomes that we're in progress and looking at are quite a few bryophytes, mosses, hornworts in collaboration with Fei Wei and group and his collaborators, some additional fern genome sequences. Within the angiosperms, we're also sequencing kind of members of the eudicots outside of the core eudicots to better understand the rapid diversification that occurred in the core eudicots. So the ancestral sequence of all core eudicots and how that genome, that hexaploid genome was sorted out among major lineages of core eudicots.
Great. Well, thanks very much. And thanks again for your talk.
Well, thanks for inviting me to participate.
Yep, my pleasure. Okay, so our next talk is by Jill Wegerson from the University of Connecticut in the US. And her talk is entitled Plant Megagenomes, Addressing Challenges in Assembly, Annotation, and Cyber Infrastructure.
All right. I just want to thank the organizers for the invitation to speak with you today. I'm going to be talking about big data in the context of big trees. So I'm going to start with giving a bit of a background and status on the conifer assembly and annotation projects. Then I'm going to move into the process of obtaining a complete genome, and then talk a little bit about some of our annotation challenges and solutions in context of these large genomes. And so just to give you a little bit of context on how large these genomes actually are, we have a whole range of genome sizes estimated here, from viral genomes on the far side, the human genome at about 3 gigabases, giant sequoia, which is one of our smaller genomes, conifer genomes that we've recently sequenced at 8 gigabases, coast redwood close to 27, and that's compared to one of the largest genomes that's been sequenced, about the exodal genome at about 30 gigabases, but we've already heard about some larger genomes this week. And so just to remind everyone, in addition to how large these genomes are, plant genomes are also repetitive. The conifers don't even fit on this chart. They are estimated to be between about 85 and 95% repetitive. You see genome size relative to repetitiveness of our plant species in red and our vertebrates in blue. So we've got a couple of challenges when we work with these large conifer genomes. And so just to take you a little bit back in time, the first conifer sequencing project was funded by the USDA and led by University of California, Davis. And the original call was for a single loblolly pine genome. This is an important paper pulp crop in the southeastern United States. But we decided to try at 2 other conifer genomes at the same time. So we included sugar pine, a species that is of ecological concern and susceptible to white pine blister rust in the western United States, as well as Douglas fir, which has economic importance in the Northwest United States and Canada. And all 3 of these conifers were some of the first to be sequenced. And our goals at that time were to develop community resources, obtain some general conifer biology knowledge, also develop new sequencing approaches to handle these large and complex genomes, as well as informatic tools. And that's really where our group came in. And I should also mention at the same time that 3 conifers were being sequenced, there were also 2 other projects underway, the Norway Spruce Project in Sweden and White Spruce funded by Genome Canada. And so we had one advantage of working with these genomes, and that was the availability of the haploid megagametophyte, and that was the primary source for a lot of our early libraries. And in fact, we really tried to maximize the use of that haploid tissue when developing the sequencing strategy. And so the sequencing strategy at that time, given the cost of sequencing and the size of the genomes, was to mostly leverage that short read sequencing. And it was done in a multiple insert style approach. And once we leveraged everything we could from the megagametophyte, and the amount of material one could obtain depending on the species varied a bit, because there is size variation there in obtaining that material, is that we then moved into using the diploid needle tissue as the primary resource for, say, mate pair libraries, which we don't hear about as much today. But also at that time, there was phosphate dye tags being used, back-end sequencing, a variety of different techniques to obtain that scaffolding a secondary step. And so with that deep short read sequencing estimated at about over 100x for most of the projects, the plan was sort of to proceed in a traditional route, take all of that short, deep sequencing, form contigs, present that to the assembler to form contigs, and then scaffold those contigs into larger pieces. And that's where those mate pair libraries or those dye tags might come into play at that stage. And so that all sounded like a good plan, except for the fact that we had over 100x in short read sequencing, and these genomes are very large. And so the Mazurka assembler was actually one of the first informatic tools developed out of the Conifer project and has remained a huge contribution to the community. This was led by Alexei Zimin at University of Maryland, and the approach essentially took all of that deep short read sequence and it reduced it by 2 to 3-fold through the formation of super reads. So it leveraged the overlap of those short reads, extended them out, error corrected them, created larger reads that can be presented to a more traditional overlap layout consensus method, which is leveraging the original Celera assembler. And that could then eventually put together high quality, more contiguous, more accurate assemblies. Because it is leveraging that overlap layout consensus step, it does take a little while to run. So it takes about 1 terabyte of RAM and it'll run for about 3 months on a 20 gigabase assembly. Newer iterations of Mazurka can leverage other applications downstream, such as SOAP de novo after that super read phase. And also even more recent iterations can now ideally leverage both PacBio and Oxford Nanopore data and really continues to produce those high quality assemblies by still leveraging a more optimized overlap layout consensus approach. And so combined with the sequencing strategy and the informatic tools, we were able to generate, in at least these first 3 assemblies that we worked on, the first references for these conifers. Now, when you look at those numbers, they're still really fragmented, right? And they're not particularly contiguous. And even that is despite a couple rounds of additional work later on, such as very, very low coverage PacBio for Labile pine, which is the Pinus taeda listed first, or the addition of 10x coverage for sugar pine, which did increase the contiguity, but we're still a long ways away from those 12 or 13 pseudomolecules that we might expect to get out of a conifer genome. And so fast forward just a little bit, we've got 4 other genomes that have come on the scene. So we have Siberian larch, we have silver fir. Both of those are still both short read assemblies, but then 2 other conifer genomes, so giant sequoia and coast redwood, that our group has been involved in. And both of those have the goal of trying to get to that chromosome scale assembly for conifers for the first time. And so both of these species were funded by the Save the Redwoods League. These are species that many people are very familiar with. They're very iconic, especially in North America. So the group was interested in restoration goals for coast redwood. Much of its natural habitat is under threat still. And then in terms of giant sequoia, where most of the habitat is preserved, there's an interest in figuring out better strategies for conservation. So now one of the challenges, however, with coast redwoods, it is a hexaploid, which is rare amongst our conifers. And then the giant sequoia is our smaller species. So that's our A gigabase genome. And so a lot of this work was led by Alison Scott, a postdoc at UC Davis. And the original approach followed very much the same plan as we did in the beginning of those first stage conifers. So we tried to get most of our sequence out of that megagametophyte tissue, very deep Illumina short read sequence. And then we went and then obtained long read sequencing. So this is deep nanopore sequencing, about 22x coverage from diploid needle tissue, and then moved all of that into high C for the first time. And this was done for both giant sequoia and a coast redwood. And so this paper just came out in G3 talking about the giant sequoia genome. So you go check it out. And this type of figure is probably old hat to a lot of people who are watching this. But for us, it was very exciting to have 11 pseudomolecules that were assembled for this giant sequoia genome and to be able to present that as a very contiguous assembly to the community. So now that we've gotten things somewhat under control in regards to sequencing strategies, great tools for assembly, getting more contiguous genomes and the resources that are out there, our group has very much been focused on genome annotation. And when we talk about that, I'm very much focused on protein coding annotation here and leveraging both structural and functional information in order to improve the annotation. And so just to remind you, there's a lot of challenges facing these genomes. So we've got the fragmentation, we've got the large size, we have the fact that their pseudogenes are very prevalent in the genome, and we also have these very uncharacteristic gene structures. So for example, in the giant sequoia paper that just came out, we identified introns that were over a megabase in size. And so that doesn't play particularly well with a lot of the tools that are out there for genome annotation. And so this just gives you kind of an idea based on some of the genomes we've been working with, including giant sequoia recently, as to the number of genes that are initially predicted out of a lot of our standard pipelines. So well over 250,000, up to 1.4 million. And we know that we're aiming to get probably somewhere between 30 and 45,000 genes in a conifer. We don't have any recent whole genome duplications, and we know that there's just not that many more genes, and there's a whole lot of repeats. So we're trying to figure out how to get to that high quality gene set in the most straightforward manner possible. So our annotation workflows is a lot of what we've worked on and focused on in the group. And a lot of those efforts have been start out much the same. We've got a soft masking approach. We are using deep RNA sequencing. We're using PacBio IsoSeq to get high quality proteins. We're aligning those resources to the genome. And we're using some of the early prediction tools, things like Breaker or Maker, to generate those base predictions. But then after that, we've developed a couple tools custom in the lab in order to improve and filter those genome annotations after that step. And so I just want to briefly introduce a couple of those. I won't have time to go to them in detail, but GFAX is one of our tools that operates on both the genome and the annotation file. It has various ways to look and identify structural challenges, deal with overlapping reads, provides visualization, and a lot of different file format conversion for downstream analysis of your genome annotations. This package can actually work in conjunction with NTAP, which is the eukaryotic non-model transcriptome annotation pipeline, can be used on both transcriptomes or gene models, and it integrates phylogenetic information, contaminant screening into the pipeline, again, to kind of feed back into determining true gene models in a genome annotation. And these packages can work together very efficiently to improve the annotation and reduce all of those false positives down. Given that, it's still a huge challenge. This is a view of kind of our BUSCO estimates. So these are your single copy orthologs that we're estimating here. And we know that our genomes are not particularly well represented in BUSCO databases yet, but nonetheless, something that you'll note is the BUSCO scores are very low at the genome level, probably a lot to do with trying to bypass those long introns. But then amongst the genomes that we've been working on and improving, we're starting to see those BUSCO scores creep up over 50%, but still lower than what we would estimate out of our transcriptomes alone. And so we're now starting to also leverage things like ATAC-seq to help us with our monoexonics and a lot of the other challenges around genome annotation. And that's just a preview of some of the work that we're going to be doing and to sort of integrate that information into our annotation pipelines. I should mention that a lot of this work is led by Samara Zalmans, who's a PhD student in our lab, and the ATAC-seq in conjunction with North Carolina State University. And with that, I want to thank so many people for participating and sharing data in these projects. We're so many different resources, transcriptome and genomic and otherwise, that led to the success of the conifer genomes. And with that, I will take questions. Thank you.
Thanks, Jill. That was really a great talk and a great overview of some of the challenges and some of the solutions that you're developing. We'll have time for one question. And this question is, with the very large introns, how much reduction in 3' exon transcription do you see?
Ooh, that's a good question. We haven't really looked into that yet. We're just sort of trying to figure out where the bounds are on these transcripts. But that's an excellent question. So absolutely, we'll look into that in the future.
Okay, great, thanks. I think we actually need to move on to the next talk. So thanks again very much.
Hello, my name is Morgan Gostel, and I'm Director of the Global Genome Initiative for Gardens. Today, I'm going to be talking a little bit about this international partnership and how it was designed to address critical needs in the biodiversity genomics community. Since this program was founded in 2015, we have sought to leverage the enormous diversity available in living collections from botanic gardens around the world to provide and enable ambitious sequencing projects in the biodiversity genomic community. I was struck when I joined GGI Gardens, or GGI, in 2015 by a quote from our director, John Coddington, who states, "The fundamental limiting factor for biodiversity genomic research is no longer the cost of sequencing, but rather the availability or accessibility of high quality or genome quality tissues." Our program set out to overcome this challenge by uniting collections with genome scale data to achieve phylogenetically informed sampling gaps on the plant tree of life. We have tried to align these priorities over time with those of the biodiversity genomics user community, such as large and ambitious projects, including the International Barcode of Life, Kew's Plant and Fungal Tree of Life program, the Earth Biogenome Project, and the 1,000 Plant Transcriptome and 10,000 Plant Genome Project. Our first order of business then was to assess levels of diversity in botanic gardens. And from the outset, we estimated, without any a priori knowledge, that botanic gardens worldwide would contain all families, 50% of genera, and about 25% of species of vascular plants around the world. We used these estimates to establish goals for the program, and these were divided into 3 phases. First, collect and sample all families. Second, 50% of all genera. And third, as a moonshot, collect and preserve genome quality tissues from all species of vascular plants on Earth. Now, coincidentally, in 2017, a paper was published by Ross Mounts and colleagues who, in collaboration with Botanic Gardens Conservation International, used the plant search database from BGCI to assess levels of diversity in these living collections from gardens around the world. What we find is that our estimates were really not far off the mark. In fact, from the 1,100 gardens in their plant search database at the time, they found that worldwide gardens contain about 30% of species, almost 60% of genera, and 75% of families of vascular plants around the world. With this collection knowledge in hand, the next step was logically to link and make accessible data about genomic tissue collections. And we're able to make this possible through a collaboration with the Global Genome Biodiversity Network. GGBN is an international network of biorepositories that store genomic tissues, their samples, and then they share information about these tissues online, similar in a way to how GenBank does for sequence data. So far, this network encompasses 95 members in 30 countries and over 1.6 million samples and growing. Now, when we talk about establishing a Global Genome Initiative for Gardens program, what we're really talking about is putting boots on the ground in a botanic garden to make voucher collections. These voucher collections are deposited in a recognized herbarium, and the genomic tissues that are sampled are then stored in a GGBN partnered biorepository. In order to make this possible, we need to do a couple of things first. One, we need to prioritize our collections from that botanic garden. We do this by performing a gap analysis. This allows us to submit an inventory of plants in the botanic garden where we're collecting, and then retrieve data about which of those samples or which of those taxa have not yet been collected and are available in GGBN. On the resources side, we are able to have teams of usually 2 to 3 individuals that include maybe a supervisor, interns, volunteers, or students, and they collect using standard herbarium and genetic tissue collection procedures. This includes a plant press, and then DNA-specific collection equipment, such as a liquid nitrogen dewar and silica gel for rapid desiccation and preservation of tissues. To date, we have developed a couple of tools to make this process more simple. This also means that gardens and biorepositories can carry out these tasks independent of GGI. So these are publicly available resources that GGI and BGCI have developed to make available to the user community. The first of these is the GGI gap analysis tool. This is a website at globalgeno.me where a user can submit a list of taxa from maybe a Botanic Gardens living collection inventory, and it will return to you information about whether that taxonomic name is already available in the GGBN database, whether or not it's available in GenBank, and then more detailed information about whether or not a DNA barcode has been sequenced for it or not. It can then prioritize the importance of the collection of that sample. To date, GGI Gardens since 2015 has collected over 400 vascular plant families and genomic quality tissue from over 4,500 vascular plant genera. Now, getting to our three-phased goals of all families, 50% of genera, and all species is going to take a lot more work. We have over 4,500 genera already, but that means that there are at least about 10,000 genera still missing from our collection. So we're only about a third of the way to our 50% of genera target. We've partnered with BGCI to leverage information in their plant search database to help us strategically target our sampling goals. We use plant search in order to compare the list of taxa that have not yet been sampled in GGBN. Those tissues are not yet available in any public genome quality tissue database, with information from BGCI's plant search to see where those missing taxa may be able to be sampled from botanic gardens around the world. And as you see in this Venn diagram, of the approximately 9,300 genera that we still have yet to sample for GGI Gardens, about 4,300 of them are still available to be sampled in botanic gardens around the world. So these gap analysis tools are proving extremely useful for not only prioritizing, but really targeting our collecting activities and where we invest our resources. So if you know of a garden that has a lot of botanical diversity, they perhaps already carry out activities like herbarium and genetic tissue vouchering, I encourage you to please reach out to them and suggest that perhaps they talk with us about joining the GGI Gardens program. And there are a number of reasons why. We provide a number of benefits to garden partners. Some of these include not only providing capacity in terms of the resources, the tools, the materials to make the collection, but this also provides unique opportunities for education and outreach for gardens, as well as linking with other projects like citizen science initiatives. Most importantly, we have just announced a new collaborative grant proposal that has a deadline of November 15th with BGCI. And this provides up to $4,500 to support voucher collection at Botanic Gardens. I encourage you to share this information with your colleagues at Botanic Gardens or reach out to us if you have any questions. With that, thank you very much. And I'd like to say none of this work could have been possible without Vicki Funk. Sadly, Vicki passed away last October after a fight with a very aggressive cancer. This entire program was the brainchild of Vicki Funk. And it was only because of her generosity and enormity of vision that this has been possible. So thank you, Vicki. This will live on, and you will live on through programs like this that you help spearhead. Many others to thank, of course, at the Global Genome Initiative, GGBN, and all of our partners, the staff, the volunteers, the interns, and the students. Thank you.
Thanks very much, Morgan. That was a great overview of all of the efforts that you've been doing for the last several years. So we have time for one question. So I'd like to ask you that given the huge number of species of angiosperms in particular, somewhere maybe approaching or exceeding 400,000 species, what needs to be done to enlarge the number of samples for genome sequencing? Either through additional efforts with botanical gardens or through coordinated efforts of field collecting?
Great question. Obviously, botanic gardens, based on the data that we have right now, only hold about 30% of species. So getting to that moonshot target of all species is only going to be possible through larger scale field collaboration. I think it, for me, really drives home how important it is that we focus on investing resources and doing field work around the world in biodiverse countries. We build collaborations beyond botanic gardens and focus in situ. So this is something that a number of initiatives have proposed for some time. One of those is a call to action from 2017 in a recent Nature paper. It's just the importance of getting boots on the ground, basic field targeting of large-scale collection programs. So I hope that we can sort of marry these ideas of focusing on things that are already in collections and then use that information to highlight what we don't have and go out and prioritize its collection.
Great, thank you very much. And thanks again for a great talk. So we need to move on. Yep, we need to move on to our next speaker. And the next talk is by Elena Maleshko from the NTNU University Museum in Trondheim, Norway. And the title of her talk is Studying the Implications of Gene Flow for Species Diversification Using Phylogenomics in Peat Moss.
Hi, I'm Lena. I'm a PhD student at NTNU University Museum in Trondheim, Norway. I'll be talking about studying the implications of gene flow for species diversification using phylogenomics in peat moss. The relative importance of introgression for diversification has long been a highly disputed topic in speciation research and remains an open question despite the great attention it has received over the past decades. Studying this question, however, is a challenging task. Many methods used to quantify interspecific gene flow require knowledge about the true phylogenetic relationship among the species. In turn, gene flow obstructs reconstruction of this relationship, thereby creating a vicious circle. Incomplete lineage sorting, or ILS, is a phenomenon when an individual of a population of a species failed to coalesce with another individual from this population, but instead coalesce with individual from another species, as shown on this picture. ILS among the diversifying species distorts phylogenetic signal in a manner similar to that of gene flow, which makes species pre-reconstruction even more difficult. Extensive ILS often occurs during rapid diversification. So disentangling the effects of gene flow and ILS require a good study model. Here we use phylogenetic methods in a bryophyte genus of peat moss, Sphagnum, for this purpose. Sphagnum has undergone recently rapid radiation and includes many species that actively hybridize with each other. We sampled in total 190 individuals from 12 commonly occurring species at 3 geographical scales in Norway, Austria, and Germany. We performed low-depth sequencing to obtain whole nuclear, chloroplast, and mitochondrial genomes. So first, we used Ruxamal to reconstruct species tree based on concatenated nuclear and organellar SNPs. The analysis resulted in well-resolved but topologically conflicting phylogenies, with several species clustering with different subgenera in nuclear and plastid phylogenies. In order to identify the reasons for this nuclear conflict, we performed coalescent-based analysis in Ancestral using circa 1,000 randomly sampled genes. To assess the distribution of phylogenetic signals across the genome, we also reconstructed trees in 100 kb sliding windows. The analysis in Ancestral resulted in the same species tree topology as the Ruxamal analysis, suggesting that the discordance is real. Moreover, the vast majority of individual gene and sliding window trees did not recover the species tree topology. We also found that deeper nodes, like these ones, were associated with more discordance, whereas the monophyly of the species was highly supported. Finally, the branch length is correlated with the node recovery by gene trees. The way phylogenetic signals differ among nuclear and plastid markers, as well as how discordance is distributed across the genome and across the nodes in the species tree, strongly suggests that the discordance is mainly caused by ILS. However, this analysis did not account for gene flow. Thus, we employed a suite of different tests for introgression, namely d-statistics using different outgroups, and F3 statistics, tree-mix, and a mixture analysis. In addition, we used a recently described QUBO method, which uses the distribution of internal branch lengths in triplets of species to differentiate between ILS and gene flow, causing phylogenetic discordance. Based on results of these and previous analysis, we found that levels of post-speciation introgression were very limited, and extensive genome-wide discordance is caused by ILS due to rapid radiation rather than post-speciation introgression. Additionally, our findings suggest that there has likely been introgression among ancestral populations, followed by ILS. So how does this fit in general context of speciation phylogenomics? In contrast to many actively hybridizing groups, post-speciation gene flow is not the main cause of phylogenetic discordance in sphagnum. And finally, rapid radiation creates a very similar phylogenetic pattern in bryophytes and angiosperms, which supports the suggested earlier idea of universality of evolutionary processes among land plants. I would like to thank all the collaborators on this project, and thank you for listening.
Thank you, Alina. What a great talk. I really enjoyed learning more about the peat mosses. One question that I have for you has to do with polyploidy, and polyploidy is reported in multiple species of sphagnum. And I'm wondering if it occurs among any of the species that you sampled and how you might take this into account in your studies, and if it might be responsible for some of the discordances.
Thank you for the question. No, we sampled only haploid species, exactly for this reason. So we wanted to make sure that we only sampled haploid ones. So for the ones that we sampled, the data suggests that there is not much variation in the chromosome number. So they should be haploid, all of them with 19 chromosomes. Yeah, so there are, as you said, a lot of polyploid species, and to include them to the analysis, we would need, I guess, a better genome, which is on the way. And then we would need to perform a more high-depth analysis in order to phase the genomes as well, I believe so. So that's beyond the scope of this study, but it would be really interesting to see that as well.
Great, thank you very much. And thanks again for a wonderful talk.
Thank you.
The final talk of our session is by Aglaya Shukala from the University of Vienna. And the title of her talk is A Transcriptomic Perspective of Adaptation to Different Altitudes in the Alps.
I am Aglaya Shukala, and today I will give you a transcriptomic perspective of the process of adaptation to different altitudes in an alpine plant species. Adaptation is a key process for the evolution of new species and ultimately for the evolution of biodiversity. So we are very interested in the drivers of this process, and in particular, we try to get insights into the process from an alpine plant species, Angiosperma pusillum, which forms two ecotypes, one growing around 2,000 meters in the alpine range, and another ecotype growing around 1,000 meters in the mountain range. While the plants in the alpine environment grows very exposed to the sun, close to streams, the plants at the lower side grows under rocks overhangs. So there are strong ecological differences between these two sites in temperature and water, as well as light availability, that are both higher at in the alpine environment. And also these two plants are morphologically distinguishable. The alpine ecotype is glabrous, while the mountain ecotype has a dense glandular indromentum. We were able to assemble the genome of these plants. It's a relatively small genome for a plant species, 1.3 gigabases. And we have performed the assembly using long, low coverage and short, high coverage reads, so a hybrid approach. As you can see, our assembly is still highly fragmented, so it could be improved, but it still recovers 95% of the estimated genome size, and annotation worked quite well, recovering 25,000 gene models and 90% of Buccal orthologs. So we were able to use this reference genome and transcriptome data, full transcriptome data from several ecotype pairs across the geographic range of co-occurrence to investigate the process of adaptation, starting from investigating demographic history. And to make a long story short, we were able to show that these ecotypes most likely diverge in parallel several times across the geographic range of occurrence, and probably all were originated postglacially. We have cultivated 3 ecotype pairs in common garden and used whole transcriptomic data to identify genes that are differentially expressed in each ecotype pair. And then we have looked for overlaps of these genes that are differentially expressed between ecotypes. And as you can see, the overlap that we find is very mild, even if significant. And this was quite surprising. But where we find a strong overlap is at the functional level. As you can see, for example, for these 2 functions, in the genes that are higher expressed in the alpine ecotype, so the one growing in the sunny environment, we have several gene ontology terms that are related to photosynthesis and that are enriched with several genes underlying this trait, while at the lower side, where the environment is drier, we have several geoterms related to response to drought. So basically, low reproducibility of gene expression responses at the level of specific genes, but high reproducibility at the functional level. And finally, we have performed reciprocal transplantation and produced full transcriptomic data also for ecotypes grown at the lower sides and ecotypes grown at a higher side. And in the scatter plots, you can see on the x-axis changes in expression as log-fold changes between ecotypes when they are cultivated at the lower side. This is the blue, light blue dots, while the orange dots show you expression differences when they are grown at a higher side, and the purple dots are the overlap. So basically genes that are always differentially expressed regardless of the environment. And what you can see quite strikingly is that the cloud of genes that are differentially expressed at the lower side is way more dense, so way more genes diverged in expression when the 2 ecotypes are grown at a lower altitude. And in particular, we know that the mountain ecotype has changes very much its expression from side to side, indicating that probably the evolution of higher plasticity of gene expression responses might have played an important role in the adaptation to this drier and warmer mountain environment. So to conclude, today I was able to show you what insights we can gain from wool transcriptomes about the process of plant adaptation to altitudes. We could gain insights into demographic history, showing a scenario of parallel ecotype formation. We could gain insight into genomic architecture of targeted traits, finding that probably adaptive traits are highly complex polygenic traits, and indeed we see repeatable selection on functions, but not on specific genes. And finally, we could show in reciprocal transplantations that phenotypic plasticity probably plays an important role in the adaptation to lower, dry, and shadier sites. And with this, I would like to thank several people from my group and my doctorate school, you for your attention, and I'm happy to take questions.
Thank you, Aglaya. That was really a great talk. And it's always fascinating to learn what's happening or has been happening in the Alps. So much rapid radiations and so much adaptation over a short period of time. I was wondering about the transcriptomes that you used. I assume these are from leaf tissues, but then I was wondering if you have if there are other tissues that you've used, and if you have possible ideas of adaptations that you might see looking at, for example, root tissue, or even maybe some of the floral tissues.
So thank you very much for the question. So we have extracted RNA only from leaf tissue. That's something I didn't say, but exactly. And we didn't extract RNA from other tissues. Unfortunately, there was no room for this. But of course, it would be very interesting, in particular in regard to the drought response, to have root tissue, for example. But this is something that might be done in the future.
Yeah, that's exactly what I was thinking, given the precipitation differences.
Yeah, one other strong difference is the photosynthetic rates, and also these trichomes that are both traits related to leaves. So this was one of the reasons why we chose this. Yes.
Great. Well, thanks Thanks very much, and I really enjoyed your talk.
Thank you.
So that concludes our session. Thanks very much to all of our presenters, and thanks to all of you for attending and joining us. Please also consider joining the chat session to continue your conversation and to have some opportunities for perhaps further interactions with our speakers. And thanks again, everyone, for joining us.